All right, so moving on in lecture 2, which is biologic sex development. We're going to talk about the typical development we'll see in a male. And I'm starting to use the word embryo or embryonic. And so that's referring to the first 8 weeks of fetal development. So after fertilization, right, then we end up forming this little ball of cells, which is kind of weird. And then that ends up becoming the embryo, which you can't see because my mug is in the way, but then ends up looking like this at about 6 weeks. So remember we talked about this indifferent stage where all embryos have exactly the same physical parts both inside and outside. So the first 6 weeks, until the 7th week, we are in the indifferent stage. Doesn't matter whether you're destined to become male-bodied or female-bodied, you have the same parts. And there they are. All right, so to remember, we have those gonads inside, right? They're going to end up forming germ cells. And then we have ducts that form. We have those two sets of ducts, the Mullerian ducts and the Wolffian ducts. And they're sometimes given different names, which is why I have those in parentheses. But remember, Mullerian ones are the pink ones, Wolffian ones are the blue ones. And then, of course, the external genitalia is also indifferent in this stage. So we have the bump, which is the genital tubercle, the genital folds right underneath it, and then the genital swelling or labioscrotal swelling outside of it. And then either the urethral slit, also called the urogenital sinus, which is right in the middle there and has an opening in it. So what happens for typical male development? How do you end up with a penis and a scrotum and epididymis and vas deferens and all of those parts? Let's go through it. So we need to remind ourselves that you're going to start with an XY chromosome. As we remember, the X is absolutely necessary for life. That's going to allow you to make a human being. And the Y is not necessary for life, but it has some important genes on it if you're going to develop as a male. So the SRY gene, called the sex-determining region of the Y chromosome, as well as genes for spermatogenesis, as well as genes that make you a little bit taller with larger hands, feet, and teeth. Okay, so that's what the Y chromosome is bringing to the table. So what's going to happen at about 7 weeks is that SRY gene is going to turn on and it's going to send a signal. And that signal is going to tell those gonads, which have been undifferentiated. And so gonads, that's the term like kick them in the nads. That's where that comes from, nads or gonads. They end up becoming testicles. And so they do a couple things once they decide to become testicles. One is they start making these baby sperm called spermatogonia. But more important for our talk today, they're going to make two different types of cells. Sertoli cells and Leydig cells are going to begin to form in those testes, in those testicles. They're going to be important. They're going to be important because those cells are going to start making hormones, and it's specific each to one. So the Leydig cells are going to start making testosterone. So you can see I made some little rectangles that are Leydig cells, and the little T's are testosterone that they're making and secreting into the body. And the Sertoli cells, with a little s, are going to make a hormone called MIS, which stands for Mullerian inhibiting substance. There's a little hint there. We've heard about Mullerian before, talking about those Mullerian ducts, those pink ducts. So that's what's going to happen now that the gonads have become testes. Now we've made these two different cell types, and these two different cells are making these two different hormones. And these hormones are the chemical messengers that are going to make things happen. So internally, MIS, right, Mullerian inhibiting substance, I think you see where this is going, it's going to tell those Mullerian ducts to degenerate. They're just going to break down, fall apart, be resorbed by the body. So they're going to go away. So you can see kind of the dotted line there for those pink ducts, they just go away. The testosterone, which was made by the Leydig cells, is going to tell those Wolffian ducts, don't give up, keep growing, keep developing, and they will persist and develop. Without testosterone, Wolffian ducts will degenerate and die. They need it to survive. With testosterone, they're going to grow and develop into the internal ductal system of the male, things we're going to learn about in a couple weeks, so vas deferens and seminal vesicles, all that cool stuff. So that's how we're going to get our internal development of the male. Here it is all together on one slide in case you wanted to review it. Externally, what's going to happen is that testosterone made by the Leydig cells in the testicles is going to tell that indifferent genitalia how to turn into a penis and scrotum. And it's going to do so the following way. So that genital tubercle, which I've kind of highlighted in red, is going to become the glans or head of the penis, as well as the top surface of the penis, which you can't see in this second drawing. The genital folds are going to come together and form the underside of the penis. The labioscrotal swelling, which is shown here in kind of that blue color, is going to fuse together and become the scrotum. And the urogenital sinus, which is the white part in the middle, is going to become the urethra, the tube that urine travels through, and that's going to be enclosed entirely within the shaft of the penis. So that's how that indifferent genitalia is going to grow and morph and change into typical male genitalia. So I kind of mentioned this, right? But those genital folds and the labioscrotal swellings have to come together and fuse in the midline, right? So male genitalia doesn't have two halves that are separate, right? Everything is together. So what you can see in this lower diagram here is that it starts as open, right? Those are the genital folds with that opening, and it's going to kind of zip up. Right? So as they develop, and it'll zip up so that eventually you just have that urethra from your urogenital sinus on the inside, that tube for the urine to travel out and semen too. But then it should be totally closed on the underside. Same thing with the scrotum. Normally it should kind of all zip up and fuse and close at the midline in the middle there. So here's that summary again of how that external genitalia is going to transform from that indifferent genitalia into the penis and scrotum. Later in fetal life, so toward the end of pregnancy, or sometimes even in the first months after birth, what's going to happen is those testicles, they actually started up near your kidneys in your body, and your kidneys are kind of up here, right, up in your back. So they're going to actually move down gradually throughout the pregnancy, down, as you can see, it's kind of this blue structure, starts up here, and it's going to move down, 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 and eventually down all the way into the scrotum around the time of birth or soon after. So that's called testicular descent, because descend means to go down. So in summary, typical male development, you have XY chromosomes, and on that Y chromosome, you have that SRY gene. The SRY gene tells the gonads to become testes, and therefore you get Sertoli cells and Leydig cells produced. The Sertoli cells make MIS, that Mullerian inhibiting substance, which causes the Mullerian ducts, the pink ones, to disintegrate and go away. The Leydig cells make testosterone, and that's going to do two things. Internally, it's going to tell the Wolffian ducts to persist and develop into the internal tubes and glands of the male reproductive system. And externally, it's going to signal that genitalia to masculinize and form a penis and scrotum. And that's it. So the next part, 2C, will be typical female development.